Welcome to BioBits and Bytes. Today you're joining us for a fascinating exploration into your body's incredible defense system, immunology.
That's right.
We're going to take a deep dive into how your body recognizes and tackles the constant stream of invaders it encounters, drawing our insights from the comprehensive Reut's Essential Immunology. Think of this as your express route to understanding the key players and strategies that keep you healthy.
Exactly. And our mission today is really to unpack the dynamic relationship between your innate and adaptive immune systems.
Okay.
We'll see how these two work in concert, not just to fend off immediate threats, but also, crucially, to build a lasting shield against future encounters. You've shared a rich collection of information, and we're eager to extract the most crucial and captivating nuggets for you.
And to make sure we're all on the same page as we navigate this, well, intricate landscape, we'll be sure to define any scientific terms that pop up.
Good idea.
So let's jump straight in. We often talk about immunity having two main arms. What are these? And, in broad strokes, what's the key difference between them?
Great question to start. So the immune system operates with two principal branches: the innate immune system and the adaptive immune system.
Right.
The innate immune system is your body's swift first responder. Think of it as the ever-vigilant security detail, always ready to react instantly to any sign of trouble.
Okay, the first line of defense. So how would you formally define innate immunity?
Okay, so innate immunity encompasses the non-specific defense mechanisms you're born with. They provide immediate protection against a really wide array of potential threats.
Immediate response, that makes intuitive sense.
And the other arm, the adaptive one.
Right, the other arm is the adaptive immune system. This one is a bit slower off the mark initially, sometimes taking days or even weeks to fully mobilize.
Slower.
But its strength lies in its remarkable precision and its ability to learn and remember past adversaries. So could you define adaptive immunity for us? Sure. Adaptive immunity is essentially a highly specific defense system that develops over your lifetime in response to encounters with particular antigens. It involves activating specialized immune cells, lymphocytes, and crucially, it creates immunological memory that offers long-lasting protection against specific pathogens.
Exactly. So think of the innate system as the general first responders, while the adaptive system is like the specialized, highly trained unit that remembers exactly how to deal with a specific threat in the future.
Okay, so innate is the rapid, general response, and adaptive is the slower, targeted, and memory-equipped force. Let's delve a bit deeper into this innate system then. How does it even know something's wrong in the first place? It can't be tailored to every single bug out there, surely.
That's a really insightful point. No, it can't. The innate immune system relies on these clever recognition molecules called pattern recognition receptors, or PRRs for short.
PRRs. Okay, define pattern recognition receptor for us.
Right. A pattern recognition receptor, or PRR, is a protein found on or within your immune cells that has the ability to detect these conserved molecular structures that are characteristic of pathogens, but generally absent from your own cells. They act like the innate system's early warning sensors.
Molecular structures of pathogens. Like what? Give me an example.
Exactly. These PRRs recognize what we call pathogen-associated molecular patterns, or PMPs.
PMPs.
Damn the definition.
Okay, pathogen-associated molecular patterns, or PMPs, are conserved molecular features commonly found on the surface or inside microbes. Things like bacterial cell wall components or viral genetic material, but they're not typically found on your own body's cells.
Got it.
Think of them as the universal danger signals of the microbial world, like a specific type of flag that many different bacteria or viruses might fly.
Danger signals. That's a good analogy. So the PRRs spot these danger signals. Are there any other kinds of trouble signs the innate system looks out for, maybe from our own cells? Yes, absolutely. The innate immune system is also very good at detecting signs of cellular damage within your own body. It does this by recognizing damage-associated molecular patterns, or DAMPs.
DAMPs. Okay, define that one too.
So a damage-associated molecular pattern, or DAMP, is a molecule released by your own damaged or stressed cells, things that shouldn't be outside the cell normally. These can also be recognized by those pattern recognition receptors we just talked about, triggering an inflammatory response.
Ah, okay.
These DAMPs act like an internal alarm indicating tissue injury, regardless of whether a pathogen is involved.
So PAMPs signal foreign invader, and DAMPs signal something's gone wrong internally. That's a smart two-pronged detection strategy. Now, you mentioned pairs aren't just one type of detector. You said there were different classes.
Yes, what's fascinating here is that pairs come in various classes, each specialized to recognize different kinds of PAMPs and strategically positioned in different cellular compartments.
Different location?
Exactly. This allows the innate immune system to generate a kind of molecular signature of the threat, providing crucial clues about the type and location of the infection.
A molecular signature that sounds like it could really help shape the initial response. Tell me more about these different PRR classes.
Certainly, one important class is the toll-like receptors, or TLRs.
TLRs. Let's get the definition.
Right. Toll-like receptors, or TLRs, are a family of receptor proteins. Many span the cell membrane, and they're found on the surface of, and also within immune cells.
Okay.
They recognize a wide variety of PAMPs, like bacterial lipopolysaccharide, that's a major component of some bacterial cell walls, and also things like viral double-stranded RNA.
So they catch different kinds of threats.
Yes, different TLRs are specific for different PMPs and are located either on the cell surface to detect extracellular pathogens or in internal compartments called endosomes to detect pathogens that have been engulfed by the cell. Think of them as different types of security cameras, each looking for a specific type of intruder in a specific location.
Okay, TLRs on the lookout on the surface and inside. What other detectors are part of this innate system?
Another key class is the C-type lectin receptors, or CLRs.
CLRs, definitely.
C-type lectin receptors, or CLRs, are a diverse family of receptors found on the cell surface that primarily recognize carbohydrate components. You know, sugar-based molecules found on the surface of many pathogens, particularly fungi and some bacteria.
Carbohydrate specialists, got it. And I recall you mentioning some that operate inside the cell as well, the cytoplasm.
Exactly. We also have the NOD-like receptors, or NLR.
NLRs, define those.
NOD-like receptors, or NLRs, are a family of pattern recognition receptors that reside inside the cell in the cytoplasm. They detect bacterial components and also signs of cellular stress that make their way into the cell's interior. Activation of NLRs can trigger inflammation.
Okay, and one more type.
And finally, there are cytoplasmic DNA sensors.
Define those two, please.
Cytoplasmic DNA sensors are a group of receptors located in the cell's cytoplasm that specifically recognize foreign DNA. This is significant because DNA shouldn't typically be floating around freely in the cytoplasm, so its presence can be a strong indicator of viral or bacterial infection.
So a whole network of detectors, each with its own specialty and location, working together to identify the threat and even provide hints about what kind of threat it is. That's quite a sophisticated initial defense. Once these threats are detected, what happens next? How does the innate system actually fight back?
Right, detection is just the first step. The innate immune system has a range of cellular and chemical weapons at its disposal. Key among these are signaling molecules called cytokines and chemokines.
Okay, cytokines and chemokines. Let's define those. First, cytokines.
Okay. Cytokines are a really diverse group of proteins that act as messenger molecules in the immune system, facilitating communication between different immune cells. They have what we call pleiotropic effect.
Pleiotropic, meaning
Meaning a single cytokine can actually have multiple different actions. Things like activating other immune cells, inducing them to differentiate into specialized fighters, or enhancing their ability to kill microbes.
got it. And chemokines. Definition.
Chemokines are another class of signaling proteins that primarily act as chemotactic factors.
Chemotactic factors, meaning they attract cells.
Exactly. They're molecules that have the ability to attract cells towards a specific location. In the immune system, chemokines are released to guide immune cells to the precise site of infection or tissue damage. Think of them as the GPS for immune cells, telling them where to go.
So cytokines are the general communicators, and chemokines are the GPS guiding the immune cells to where they're most needed. How are these released, and how do they exert their effects?
These messenger proteins are typically released by immune cells in direct response to detecting those PAMPs and DAMPs we discussed earlier. Once released, they diffuse away and bind to specific receptor proteins on the surface, the plasma membrane of other cells.
Right.
This binding then triggers a cascade of events inside the receiving cell, known as intracellular signaling pathways, which ultimately alters the cell's activation state and its behavior, essentially rallying it to join the ongoing immune response.
Okay, the chemical signals are out. Now, what about the actual boots on the ground? Which cells are the main players in innate immunity, and how do they go about eliminating the threats?
The primary cellular players in innate immunity are phagocytic cells like macrophages and neutrophils.
Okay, macrophages and neutrophils.
Macrophages are kind of like resident guards. They're found in tissues throughout your body, acting as sentinels. Neutrophils, on the other hand, are recruited in large numbers from the bloodstream to sites of infection when needed.
And they both eat pathogens, phagocytosis.
Exactly. Both of these cell types are capable of phagocytosis, which is the process of engulfing and essentially eating pathogens or cellular debris.
And how do they actually destroy these pathogens once they've been swallowed? It's not just digestion, is it?
No, it's much more aggressive than that. They employ several powerful microbicidal mechanisms. One crucial method involves the production of reactive oxygen intermediates.
Reactive oxygen intermediates, like bleach.
Sort of. They're chemically reactive molecules containing oxygen, such as superoxide, hydrogen peroxide, hydroxyl radical, and even hypochlorous acid. Yes, the active ingredient in bleach, but produced in a controlled way inside the cell. These molecules are highly toxic to microorganisms.
Wow. And how is this made?
This production is driven by an enzyme complex called NADPH oxidase.
NADPH oxidase.
This enzyme facilitates the transfer of electrons from a molecule called NADPH to molecular oxygen, generating superoxide, which is then converted into those other reactive oxygen species. The activation of NADPH oxidase itself involves complex signaling pathways that get triggered when the phagocytic cell engulfs a pathogen.
So essentially, they're creating toxic oxygen compounds inside the cell to kill the invaders. What other weapons do these phagocytes have in their arsenal?
They also generate nitric oxide, or NO.
Nitric oxide. Isn't that involved in blood vessel dilation?
It is, but it also acts as a reactive nitrogen species with potent antimicrobial properties. It can even combine with superoxide to form an even more toxic molecule called peroxynitrite. The enzyme responsible for nitric oxide production is actually structurally similar to NADPH oxidase.
Okay.
Nitric oxide and its byproducts can damage microbial proteins, DNA, and mess with essential components like iron. And finally, there are also oxygen-independent antimicrobial systems, things like lysozyme, which breaks down bacterial cell walls, and various antimicrobial peptides that directly punch holes in microbial membranes.
Yeah, a real chemical warfare happening inside these cells. Now, you also mentioned the complement system earlier. What exactly is that, and how does it contribute to innate immunity?
The complement system is another vital part of innate immunity. It's not a single molecule, but a cascade involving a series of plasma proteins that circulate in your blood in an inactive state. They can be activated directly by the surfaces of microbes, leading to a range of powerful effector functions. This includes directly bursting open pathogens, lysis, marking them for easier engulfment by phagocytes, that's opsonization, and also promoting inflammation to recruit more help.
That sounds powerful. How does this complement system know to attack microbes and not our own healthy cells? That seems crucial.
Absolutely critical. One of the main ways complement gets activated specifically by microbes is through what's known as the alternative complement pathway. This pathway is actually sort of spontaneously ticking over at a low level all the time.
Really?
Yes, but its amplification is very tightly controlled on your own body's cells. On microbial surfaces, however, a key complement protein called C3b can bind to another protein called factor B. This complex gets cleaved by another factor, factor D, to create an enzyme called the C3 convertase, specifically C3BB.
C3BBB, the convertase.
Right. And this C3 convertase acts like an engine, chopping up lots more C3 protein into C3A, which promotes inflammation, and more C3b. This C3b can then form more convertase, leading to a rapid amplification loop right there on the microbe surface.
So C3BB is the engine driving this pathway on the microbial surface. What prevents it from running wild on our own cells then?
That's where regulation comes in. Your own cells express specific regulatory proteins like factor H and factor I. When C3b happens to land on one of your own cells, it binds much more strongly to factor H than to factor B.
Okay.
Factor H then recruits factor I, which basically inactivates the C3b, preventing that C3 convertase enzyme from forming or persisting on your tissues. So your cells actively protect themselves.
Clara.
In contrast, on many microbial surfaces, C3b tends to bind factor B more strongly, or the surface lacks those regulators, making it somewhat unprotected. Plus, another protein called properdin can come along and stabilize the C3BB convertase on the microbe, ensuring the complement attack continues where it's needed.
That's a clever way to specifically target invaders while safeguarding our own tissues. You also mentioned proteins called ficolins and collectins earlier. What are these and what role do they play?
Ficolins and collectins are another important group of proteins contributing to your innate immunity. Think of them as soluble pattern recognition receptors. They circulate in your blood and other fluids and can bind to specific patterns on pathogens.
Soluble PRRs. Interesting. What do they recognize?
Specifically, they tend to recognize carbohydrate-based PMOPs on microorganisms. Phycolins, for instance, often recognize structures like N-acetylglucosamine, which is common in the cell walls of many pathogens.
And what happens when they bind?
The binding can do a couple of things. Firstly, it can activate another pathway of the complement system called the lectin pathway, providing an alternative route to trigger that cascade. Secondly, they can also act as opsonins themselves, coating the microbes to make them tastier for phagocytes.
So they enhance phagocytosis too.
Exactly. And they can even cooperate with other innate proteins like C-reactive protein, CRP, to broaden the range of bacteria recognized. Interestingly, the recognition ranges of phycolins and another key colectin, mannose-binding lectin, MBL, are somewhat complementary, covering a wider spectrum of microbial sugars together. It shows how the system has evolved multiple ways to spot these common microbial signatures.
It's fascinating how many layers and intricate strategies the innate immune system has. Now, let's shift our focus to the adaptive immune system. You mentioned its key characteristics are specificity and memory. How do T cells and B cells contribute to these crucial features?
Right. The adaptive immune response is orchestrated primarily by two main types of lymphocytes. T lymphocytes, or T cells, and B lymphocytes, or B cells.
Okay, T cells and B cells. Let's define T lymphocytes first.
Sure, T lymphocytes, or T cells, are a type of white blood cell that matures in an organ called the thymus, which sits in your chest. They play a central role in what we call cell-mediated immunity. This involves things like directly attacking infected cells, helping other immune cells function better, and regulating the overall immune response.
Okay, direct action and regulation. And B lymphocytes, definition.
B lymphocytes, or B cells, are another type of white blood cell, but these mature in the bone marrow. Their primary role is to produce antibodies, which circulate in the blood and other fluids. This provides what we call humoral immunity.
So T cells are more about cell-to-cell interactions and regulation, while B cells are the antibody-producing factories. And these are the cells that give us that remarkable specificity and long-term memory, correct?
Precisely. Both T cells and B cells are highly antigen-specific. An antigen, remember, is any molecule the immune system can recognize. Each individual T cell or B cell has a unique receptor on its surface that can recognize a very specific part of a pathogen, a specific antigen.
One cell, one target, essentially.
Pretty much. And furthermore, they both exhibit immunological memory. This means that upon a second or subsequent encounter with the very same antigen, they can mount a much faster, stronger, and more effective immune response. This ability to remember past infections is the fundamental basis of long-term immunity, and of course, the principle behind how vaccines work.
How do these cells develop their incredibly specific recognition abilities?
Yeah.
You mentioned T cells mature in the thymus. What are their main roles once they become mature?
T cells start as precursors in the bone marrow but complete their development in the thymus. During this maturation, they acquire unique T cell receptors, or TCRs, on their surface.
DCRs.
These TCRs are the molecules that recognize specific antigens. But there's a key difference from B cells. T cells typically recognize antigens that have been broken down into small fragments, peptides, and are then presented to them on the surface of other cells by special molecules called major histocompatibility complex, or MHC molecule.
MHC. We'll definitely come back to those. So what types of T cells are there?
There are several major types. Helper T cells, often CD4 plus T cells, are crucial for coordinating the immune response. They help B cells make better antibodies and activate macrophages to kill microbes more effectively. Regulatory T cells, TREGs, are essential for putting the brakes on immune responses, preventing excessive inflammation and autoimmunity. And cytotoxic T cells, usually CD8 plus T cells, are the killer cells. They directly recognize and destroy cells infected with viruses or other intracellular pathogens, and also some cancer cells.
The diverse team. And B cells mature in the bone marrow. What's their development like, and how do they get activated to start churning out those antibodies?
B cells do their whole development in the bone marrow. Like T cells, they develop unique antigen receptors. In their case, these are membrane-bound antibodies, also called immunoglobulins or B cell receptors, BCRs.
So they're receptors in the antibody.
Exactly, just stuck to the surface initially. When an antigen enters the body, it encounters this vast library of B cells, each displaying a slightly different antibody. Only those B cells whose antibody receptors bind strongly and specifically to that particular antigen receive a crucial activating signal.
Antigen selection.
Right. This antigen selection triggers these chosen B cells to activate and develop into one of two main cell types. Some become plasma cells. These are basically antibody-secreting factories, pumping out large quantities of soluble antibodies with the exact same specificity as the original receptor.
The antibody factor.
And others develop into memory B cells. These are long-lived cells that remember the antigen and can mount a rapid and robust antibody response if you encounter that same threat again later. It's also important to note that helper T cells often provide critical signals needed for B cells to really ramp up proliferation and differentiate effectively into plasma cells and memory cells.
So the antigen acts like a key, selecting the specific B cells and T cells with the right lock, the matching receptor. And this leads to both immediate fighters and long-term memory cells. How do we actually know for sure that lymphocytes are the cells responsible for this crucial immunological memory? Is there experimental proof?
Oh, yes. The fact that lymphocytes carry immunological memory has been very convincingly shown through elegant experiments using a technique called adoptive transfer.
Adoptive transfer.
Yeah. Basically, you take lymphocytes from an animal that's already immune to a specific antigen, meaning it has memory cells for it. Then you transfer these lymphocytes into a genetically similar animal whose own lymphocytes have been destroyed, maybe by radiation.
Okay, like a blank slate recipient.
Exactly. This recipient now lacks its own adaptive immune system but has the transferred lymphocytes. Now, if you expose this recipient animal to the same antigen the first animal was immune to, it mounts a rapid and strong secondary immune response, the hallmark of memory. And crucially, if you expose it to a different, unrelated antigen, it only mounts a slow primary response. This shows the memory is specific to the antigen the original lymphocytes encountered. These kinds of experiments with careful controls were fundamental in proving that lymphocytes are indeed the carriers of immunological memory.
That adoptive transfer experiment really provides compelling evidence. Now, we've looked at the innate and adaptive immune system somewhat separately. How do these two powerful systems actually interact and work together? They can't operate in complete isolation, can they?
Absolutely not. They're in constant communication and collaboration. A great example is how your body deals with cells infected by viruses.
Okay, virus-infected cells.
The initial innate response involves cells called natural killer cells, or NK cells.
Natural killer cells.
Yeah.
NK cells. Define those.
Right. Natural killer NK cells are a type of cytotoxic lymphocyte, meaning they can kill other cells, but they belong to the innate immune system. They can recognize and kill virus-infected cells and some tumor cells without needing prior sensitization like T cells do.
How do they recognize them?
NK cells have what are called killer activating receptors. When these receptors encounter certain stress signals or molecules that appear on infected or cancerous cells, it triggers the NK cell to release its toxic payload and destroy the target cell. So NK cells provide an early, somewhat non-specific defense.
So NK cells are the innate system's early responders for dealing with infected cells. How does the adaptive immune system then get involved in this particular scenario?
The adaptive response brings in more targeted mechanisms like antibody-dependent cellular cytotoxicity, or ADCC, and the highly specific action of cytotoxic T cells.
ADCC, let's define that one.
Antibody-dependent cellular cytotoxicity, or ADCC, beautifully bridges the innate and adaptive arms. In ADCC, NK cells use specialized receptors on their surface, called FTC receptors, to recognize antibodies specifically, virus-specific antibodies produced by B cells that have already bound to viral antigens on the surface of an infected cell.
Ah, so the antibody acts like a flag for the NK cell.
Precisely. It tells the NK cell, this cell is infected, kill it. The NK cell then releases its cytotoxic granules and destroys the antibody tag cell, so the adaptive system directs the innate killing power.
Clever. And the T cells?
Then, the fully developed acquired response brings in the cytotoxic T cells. These T cells, as we discussed, use their T cell receptors, TCRs, to specifically recognize small fragments of viral proteins, viral peptides presented on the surface of infected cells by MHC class I molecules. Binding to this specific peptide MHC complex leads to the highly targeted killing of that infected cell.
So NK cells can either directly recognize stressed cells or be guided by antibodies, and then later the cytotoxic T cells arrive with their very precise targeting. It really sounds like the innate response is crucial for kicking off and shaping the adaptive response. Is that a fair assessment?
That's a very accurate way to put it. The innate immune system absolutely plays a critical role in triggering and shaping the adaptive response. While innate receptors provide that rapid first line of defense, they generally lack the exquisite specificity of adaptive receptors like TCRs and antibodies.
Right. Remember, the incredible specificity of TCRs and antibodies comes from that random genetic recombination during lymphocyte development. But the innate response provides the crucial early signals, cytokines, chemokines, cell activation, that tell the adaptive system, hey, there's a problem, here's what kind of problem it seems to be, and helps direct the most effective type of adaptive response, whether it should be mainly antibody-based or cell-mediated.
And this leads to memory.
Yes, and that clonal expansion of the specific T and B cells that recognize the pathogen not only clears the current infection, but also generates those vital memory cells, ready for rapid deployment if the same pathogen shows up again.
Random genetic recombination to generate such specific receptors, that sounds amazing. But let's stick with our overview for now. We've talked cells. Let's zoom in on the key molecules of the adaptive system, starting with antibodies. How do they actually work to neutralize or eliminate pathogens once they're made?
Antibodies have several key jobs. One important mechanism is triggering the classical pathway of the complement system, which we talked about earlier with innate immunity. Right, the cascade.
When multiple IgG antibody molecules cluster together on a pathogen surface, which happens when they bind antigens there, they create a docking site for the first complement component, C1q. This binding kicks off that cascade, leading ultimately to pathogen lysis, inflammation, and opsonization, marking it for phagocytosis.
And do all antibodies do this equally well?
Not quite. Different subclasses of IgG have varying abilities. IgG1 and IgG3 are generally the best at triggering complement. IgG2 can do it, but usually needs a higher density of antigens. And IgG4 typically doesn't trigger this classical pathway much at all.
Interesting variation. What other ways do antibodies help fight infection?
They also interact with various FCO receptors on the surface of different immune cells, like macrophages, neutrophils, and NK cells. Remember, FC receptors bind to the tail end, the FCO region, of the antibody molecule. When these receptors bind to antibodies already coding a pathogen, it triggers effector functions. It can strongly enhance phagocytosis, making the coded pathogen much easier for cells to engulf. It can trigger ADCC, as we discussed with NK cells, and it can also trigger the release of inflammatory mediators, helping recruit more immune cells to the fight. These FTC receptor interactions can also play regulatory roles, helping to fine-tune the immune response.
Okay, so they can flag pathogens for destruction by phagocytes and NK cells and trigger inflammation. What about IgE antibodies? They're always linked with allergies, but do they have a normal defense role?
Absolutely. While IgE is famous, or perhaps infamous, for its role in allergies, it has a crucial role in defense, particularly at body surfaces like your respiratory and digestive tracts, which are constantly exposed.
How does it work there?
IgE binds with extremely high affinity to its receptor, Fc epsilon Ri, found on mast cells and basophils. When IgE bound to these mast cells encounters its specific antigens, say, from a parasite trying to invade your gut lining, it triggers rapid degranulation.
Releasing histamine.
Exactly. Histamine and other vasoactive amines, plus cytokines and inflammatory mediators. While this causes allergy symptoms if the antigen is pollen, the likely evolutionary purpose is a rapid call to arms at sites of invasion. It helps quickly recruit plasma proteins, complement, and effector cells like neutrophils and eosinophils to deal with pathogens, especially parasites like worms that might breach other defenses like IgA. Eosinophils in particular are good at damaging antibody-coated parasites.
So IgE's role is that double-edged sword, vital for defense, but also the culprit in allergies. Now, how do antibodies get passed from mother to child? That seems critical for newborn protection.
That crucial transfer is largely mediated by the neonatal Fc receptor, or FcRn.
FcRn. Define that.
Okay. The neonatal Fc receptor, FcRn, is a specialized Fc receptor that's key for transporting IgG antibodies from the mother's circulation across the placenta to the developing fetus during pregnancy.
Okay, across the placenta.
This gives the newborn passive immunity a starting set of the mother's antibodies, offering protection during those vulnerable first months before their own immune system fully matures. FcRn might also help transport IgG from breast milk across the infant's gut.
And does it do anything else?
Yes, a really important role. FcRn is also essential for maintaining the long half-life of IgG in your blood, both as an adult and child. It basically rescues Ig from being degraded inside cells. It binds IgG in acidic compartments and recycles it back to the surface, significantly extending how long IgG antibodies stick around in your system.
So FcRn is key for both newborn immunity and IgG longevity throughout life. Let's look at the antibody structure itself. What gives them their incredible diversity to recognize so many different things?
The specificity lies in their variable regions at the tips of the Y shape. The variable part of the heavy chain is genetically encoded by joining three distinct gene segments: V variable, D diversity, and J joining. The light chain variable region uses just V and J segments.
VDJ. Okay.
Now, crucially, your DNA contains multiple different versions or alleles of each of these V, D, and J segments. During B cell development, specific V, D, and J segments are randomly selected and stitched together in a unique combination. This combinatorial diversity is a huge source of the antibody repertoire.
Just mixing and matching those segments generates massive variety.
Exactly. Plus, there's additional diversity introduced at the junctions where these segments are joined, and the genes themselves are polymorphic in the population. Some species even use a process called gene conversion to further diversify their antibody genes, copying bits from non-functional pseudogenes into the active variable region.
Fascinating genetic acrobatics. And this variable region is what actually binds the antigen. What does that look like?
Yes, the variable regions form the antigen binding sites. Antibodies can recognize specific 3D shapes on the antigen surface. These are called topographic or discontinuous epitopes, formed by amino acids that might be far apart in the linear sequence but are folded together. They can also recognize short, linear sequences of amino acids, often in flexible loops. These are continuous epitopes. Essentially, the antibody makes contact with a specific footprint on the antigen surface.
And how strong is this binding? We hear about high-affinity antibodies. What does that mean practically?
Affinity describes how tightly an antibody binds to its antigen. It's measured by constants like Ka association and KD dissociation. High affinity means a high Chi and a low KD. The antibody binds strongly and doesn't let go easily.
Low KD is tight binding.
Right. The KD is roughly the concentration of antigen needed to occupy half the antibody binding sites. High-affinity antibodies often have KD values in the nanomolar NM range. This means that even at the typical concentrations of IgG found in your blood, these antibodies can bind effectively and stably to their target antigen, which is crucial for protection, especially against reinfection.
High affinity at normal antibody levels makes sense for efficient protection. Now let's turn to T-cell receptors, TCRs. How are they similar to and different from antibodies in recognizing antigens?
TCRs share a basic structural similarity with antibodies. They're built using that same immunoglobulin fold domain structure. But a key difference is TCRs are always stuck on the T-cell surface. They're never secreted as soluble molecules like antibodies can be.
Okay, always membrane-bound. And their job?
Their main function is to probe the surfaces of other cells, looking for those small peptide fragments derived from proteins, both self and foreign, that are presented by MHC molecules.
Scanning for peptide MHC complexes.
Exactly. For a T cell to get activated, its TCR must specifically recognize a particular peptide bound to an MHC molecule. Because of the huge diversity of TCRs and the fact that most peptides presented are self-peptides, finding that perfect foreign peptide MHC match is actually a pretty rare event for any given T cell.
So TCRs recognize these processed antigen fragments on MHC. What does the interaction look like structurally? How does the TCR actually bind?
Compared to the deep pockets or grooves often found in antibody binding sites, the antigen binding sites of TCRs tend to be relatively flat.
Flatter. Why?
This flatter surface seems well suited for interacting with the somewhat undulating surface presented by the peptide nestled in the MHC groove. The TCR typically sits across the peptide MHC complex, often diagonally. Specific loops in the TCR variable regions, the CDR1 and CDR2 loops, often contact the alpha helical parts of the MHC molecule itself.
And the peptide.
The specificity for the actual peptide comes mainly from the more variable CDR3 loops of the TCR alpha and beta chains. These CDR3 loops make crucial contact with the amino acid residues of the peptides sitting in the MHC groove, especially the central ones. There's some evidence the TCR might first make a general peptide-independent contact with MHC, and then the CDR3 loops adjust for optimal peptide binding. Stable binding involving multiple TCRs triggers activation through the associated CD3 signaling complex.
And what about the CD4 and CD8 co-receptor molecules we mentioned? How do they fit in?
Ah, yes. CD4 and CD8 are crucial co-receptors. They help stabilize the T cell's interaction with the antigen-presenting cell and also contribute to signaling. CD4, found on helper T cells, binds to a non-variable part of MHC class II molecules. CD8, on cytotoxic T cells, binds to a non-variable part of MHC class I molecules.
So they bind MHC2, but not the peptide part.
Exactly. They bind to conserved regions on the MHC molecule distinct from the peptide groove. This not only strengthens the overall interaction, but also helps ensure the right type of T cell interacts with the right class of MHC CD4 T cells with MHC class II, which usually presents peptides from extracellular sources, and CD8 T cells with MHC class I, presenting peptides from intracellular sources like viruses. It directs the appropriate response.
We talked earlier about receptor editing in B cells to fix self-reactive antibodies. Does anything similar happen with T cells?
Yes, a similar process, often called TCR revision or editing, can occur during T cell development in the thymus. If a developing T cell makes a TCR that strongly recognizes a self-peptide presented by MHC, instead of just dying, it sometimes gets a second chance.
A second chance. How?
It can undergo further rearrangement of the genes encoding the alpha chain of its TCR. This secondary rearrangement might generate a new TCR specificity that is no longer self-reactive, rescuing the cell from elimination. The rules governing these rearrangements, the 1223 rule related to recombination signal sequences, RSSs, differ slightly between the alpha and beta chain genes, influencing how this editing works.
Okay, so both B and T cells have ways to try and correct self-reactive receptors. Now let's circle back to those natural killer cells. Innate cells, but with receptors. How do they decide when to kill?
NK cells are fascinating innate lymphocytes, yes. Unlike T and B cells with their one highly specific antigen receptor, NK cells integrate signals from a whole array of activating and inhibitory receptors to make that kill or don't kill decision.
Activating and inhibitory. A balance then.
Exactly. When activated, NK cells can secrete cytokines like IFNO and kill target cells using cytotoxic granules or molecules like Fas ligand, but they also have crucial inhibitory receptors that prevent them from attacking healthy host cells.
So it's not just about recognizing bad signals, but also recognizing good or normal signals to hold back.
Precisely. An NK cell is generally triggered to kill only if the signals from its activating receptors, which recognize molecules often upregulated on stressed, infected, or tumor cells, outweigh the signals from its inhibitory receptors, which typically recognize molecules that should be present on healthy cells, most notably MHC class I molecules.
So if a cell loses its MHC class I,
Then it loses a major don't kill me signal for the NK cell, making it susceptible to attack. This can happen during some viral infections, or in some tumors they try to hide from T cells by downregulating MHCI, but that makes them visible to NK cells. It's a clever system.
what are some of these activating inhibitory receptors and how do they signal?
There are several families, including C-type lectin receptors, CTLRs, and immunoglobulin-like receptors. Both families contain members that are either activating or inhibitory. The inhibitory receptors often have specific motifs in their cytoplasmic tails called items, immunoreceptor tyrosine-based inhibitory motifs.
I think
Right. When these receptors bind their ligand, like MHCI, the items get phosphorylated and recruit phosphatase enzymes that remove phosphate groups, which then shut down the activating signaling pathways inside the NK cell.
putting the brakes on and activating ones.
Activating receptors often associate with accessory proteins that have activating motifs called ITAMs, immunoreceptor tyrosine-based activation motifs in their tails. Ligand binding leads to ITAM phosphorylation, which recruits kinases enzymes that add phosphate groups, initiating signals that lead to NK cell activation and killing. The overall outcome depends on the integration of all these signals.
A really elegant balance of activating and don't kill me signals. Now, we've mentioned MHC molecules a lot, presenting peptides to T cells, inhibiting NK cells. Why are these MHC molecules so incredibly variable, so polymorphic within the human population?
The MHC region is one of the most polymorphic, meaning it has the most different versions or alleles in the entire human genome. This variation is particularly concentrated in the genes encoding the peptide binding domains of MHC class I, HLA-A-BC, and MHC class II, especially the beta chains of HLA-DR-DP-DQ.
Right, in the part that holds the peptide.
Exactly. The amino acid differences are clustered right in the floor and walls of the peptide binding groove, the very surfaces that contact the peptide, and are surveyed by T cell receptors.
And why so much variation there?
This is thought to be driven by positive selection pressure from pathogens. It's an evolutionary arms race. Different MHC alleles can bind and present a different repertoire of peptides. By having huge diversity at the population level, it ensures that the human species as a whole can present peptides from a vast array of different pathogens. If a new virus emerges, it's much more likely that at least some individuals in the population will have MHC alleles capable of presenting its peptides effectively, allowing them to mount an immune response and ensuring the survival of the species.
So population-level diversity is an insurance policy against diverse pathogens. What one person's MHC can't present well, another's might. Now, let's talk about something unusual. Superantigens. What are they and how do they mess up the system?
Superantigens are pretty disruptive. T-cell superantigens, like certain toxins made by staph bacteria, have this weird ability to bypass normal antigen processing and presentation. They can physically cross-link MHC class II molecules on an antigen-presenting cell directly to the T-cell receptor on many different T-cells, regardless of what peptide the T-cell is supposed to recognize.
They just bridge them together.
Yeah, often binding to the outside faces of both molecules. This nonspecific massive cross-linking activates a huge fraction of the T-cell population, maybe up to 20%, compared to less than 0.01% for a normal antigen.
Wow, massive activation. What's the consequence?
This leads to a huge systemic release of pro-inflammatory cytokines, a cytokine storm, which can cause symptoms like fever, shock, and widespread inflammation, as seen in toxic shock syndrome. There are also B-cell superantigens, like staph protein A or HIV GP120, that can similarly activate a large proportion of B cells nonspecifically.
So superantigens are like immune system hijackers, causing chaos without specific targeting. That sounds dangerous. Let's clarify two terms, antigenicity versus immunogenicity. They sound similar.
They do, but there's a key difference. Antigenicity is simply the ability of a molecule to be recognized by an antibody or a T-cell receptor. Can it bind? If yes, it's antigenic.
Okay, just recognition.
Immunogenicity is the ability of that molecule, when introduced into the body, to actually elicit an immune response to stimulate antibody production or T-cell activation. So it has to be recognized and trigger response.
So not all antigens are immunogens.
Exactly. Small molecules called haptens are a classic example. They can be antigenic, an antibody can bind to them, but they're usually not immunogenic on their own. They're too small to effectively cross-link receptors and activate lymphocytes. But if you chemically link a hapten to a larger carrier protein, the whole complex becomes immunogenic and you can generate antibodies specific for the hapten. Also, even on a large protein, some parts, epitopes, are more immunogenic than others, often depending on how accessible they are on the surface.
So an antigen can be recognized, but an immunogen actually provokes a response. The carrier protein makes the hapten visible enough.
Oh.
Now we've talked cells and molecules. How do these cells get around? What's the immune traffic control system?
Immune cells are constantly on the move. Their traffic is precisely regulated. Lymphocytes, for instance, migrate to specific lymphoid tissues like lymph nodes, spleen, and mucosal sites like in your gut or lungs. This homing is guided by specific interactions between homing receptors on the lymphocyte surface and addressins on the endothelial cells, lining specialized blood vessels called high endothelial venules, HEVs, in these tissues.
Like molecular zip codes?
Kind of, yeah. Chemokines and their receptors are also critical guides. For example, specific chemokines produced during lung inflammation attract T cells expressing the corresponding chemokine receptors, pulling them out of the blood and into the lung tissue.
And they circulate everywhere.
Yes, many immune cells, lymphocytes, NK cells, monocytes, neutrophils, etc., continuously circulate in the blood and lymphatic system. The endothelium lining these vessels tightly controls their exit into tissues. This constant surveillance and ability to migrate rapidly to sites of infection or injury anywhere in the body is essential for effective protection.
A continuous surveillance and rapid deployment system. Now let's revisit innate effectors. We mentioned soluble factors like complement and antimicrobial peptides. How do they directly fight infection?
Right. Besides the cells, the innate system has these soluble weapons. Complement proteins, lysozyme, which degrades bacterial walls, antimicrobial peptides, which poke holes in microbes, and lectins like MBL can directly bind pathogens. This binding can trigger the complement cascade, leading to lysis, act as opsonins to enhance phagocytosis, or directly damage the microbe. Neutrophils can also release their granule contents extracellularly and deploy nets, neutrophil extracellular traps, which are webs of DNA studded with antimicrobial proteins that physically ensnare and kill pathogens.
So, direct attack from soluble molecules and neutrophil traps. Let's focus on macrophages again. You mentioned M1 and M2 polarization. What does that mean functionally?
Macrophages are incredibly versatile. They can change their functional state based on signals they receive. This is polarization. The two main types are M1, classically activated, and M2, alternatively activated.
M1 versus M2.
M1 macrophages are typically induced by pro-inflammatory signals like IFNO from T cells or by microbial PAMPs. They become highly pro-inflammatory and microbicidal, producing cytokines like TNFA and IL-12, and are good at presenting antigens. Think of them as geared up for fighting.
The warriors.
And M2.
M2 macrophages are usually induced by anti-inflammatory cytokines like IL-4 or IL-10. They are more involved in dampening inflammation, promoting tissue repair, and wound healing. They produce anti-inflammatory cytokines like IL-10 and enzymes like arginase that can actually suppress T-cell responses.
Tissue repair and calming things down.
Right. The balance between M1 and M2 macrophages in a tissue can really influence the type of adaptive immune response that develops. M1s tend to push towards Th1 cytotoxic responses, while M2s can favor Th2 responses or suppression.
Dynamically reprogrammed based on need. Amazing adaptability. We keep mentioning cytokines. Can we get a better sense of their sheer diversity and how they work? Cytokines are truly the language of the immune system. It's a huge family: interleukins, IL-1 to IL-31+, interferons, IFNs, tumor necrosis factors, TNFs, chemokines, and others. Each is produced by specific cells under specific conditions and acts on target cells by binding to specific receptors.
That's some different messages.
Yes, and diverse effects. IL-2 drives T-cell growth. IL-4 pushes towards Th2 and IgE production. IFNO activates macrophages and promotes Th1. TNF-4 fuels inflammation. They bind to distinct receptor families: hematopoietin receptors, IFN receptors, TNF receptors, Ig superfamily receptors, chemokine receptors, which are GPCRs, TGF receptors. Binding often involves multiple receptor subunits coming together to initiate signaling inside the cell.
And chemokines are a subset.
Yes, the specialized subset for cell trafficking, classified by their cysteine arrangements, CXC, CC, etc. They guide cells to inflammation, inflammatory chemokines, or maintain normal positioning in lymphoid organs, homeostatic chemokines. They act mostly locally and transiently, and there's often redundancy, multiple cytokines doing similar things, and synergy, cytokines working together for a greater effect.
A complex dynamic communication network. Let's dive deeper into those T helper cell subsets you mentioned: Th1, Th2, Th17, TFH, TREG. What are their specialized roles?
CD4 plus T helper cells are the master orchestrators. Upon activation, they differentiate into distinct subsets with specialized functions. Th1 cells, making IFNO and IL-2, are key for activating macrophages to kill intracellular pathogens and supporting cytotoxic T-cell responses. They can even license dendritic cells to make them better at stimulating cytotoxic T-cells.
Th1 for intracellular threats.
Largely, yes. Th2 cells, making IL-4, IL-5, and IL-13, are crucial for fighting parasitic worms, helminths, and are heavily involved in allergic responses, promoting IgE and activating eosinophils.
Th2 for parasites and allergies.
Th17 cells produce IL-17 and are important for defense against extracellular bacteria and fungi, especially at mucosal surfaces, but they can also contribute to inflammatory diseases. T follicular helper, TFH cells, migrate into B cell follicles and produce IL-21. They're absolutely essential for helping B cells form germinal centers, undergo class switching, and produce high-affinity antibodies.
TFA for antibody help.
Exactly. And finally, regulatory T TREG cells expressing the transcription factor FOXP3 are the peacekeepers. They suppress immune responses, maintain tolerance to self-antigens, and prevent autoimmunity.
A specialized team for different challenges. How does the system create the memory cells that provide long-term immunity?
That's a really active area of research, exactly how a lymphocyte decides to become a long-lived memory cell versus a short-lived effector cell. Several factors seem to play a role. One idea is the strength or duration of the initial antigen signal. Perhaps weaker or shorter stimulation favors memory development.
Signal strength matters.
Possibly. Another idea involves asymmetric cell division. When an activated lymphocyte divides, it might distribute key proteins or fate determinants unevenly between the two daughter cells, programming one towards memory and the other towards effector function. Certain polarity proteins seem involved in setting this up.
Division itself could determine fate.
It's a possibility. Also, surviving the initial response is key. Upregulating anti-apoptotic molecules like CD44 on T cells, which can interfere with fast-mediated cell death, might help cells persist and enter the memory pool.
So signal strength, how they divide, and surviving the initial battle all contribute. The immune system is powerful. How is it regulated to prevent constant activation or self-attack?
Regulation is critical. One major mechanism is this neuroendocrine feedback loop. Pro-inflammatory cytokines like IL-1, IL-6, and TNF, released during an immune response, actually signal to your brain, the hypothalamus and pituitary gland.
The immune system talks to the brain.
Yes. This triggers the adrenal glands to produce glucocorticoids, like cortisol, your natural stress hormones. These glucocorticoids are powerfully immunosuppressive. They circulate and dampen down the activity of immune cells, particularly Th1 cells and macrophages, providing negative feedback to calm the response. They can even promote the development of those regulatory T cells we mentioned, further helping to restore balance.
So the body's stress response helps regulate the immune response.
Yeah.
Beautiful integration. Let's quickly recap T and B cell development. T cells in the thymus.
Right. Precursors from bone marrow go to the thymus, progress through stages, double negative, double positive, single positive CD4 or CD8. Guided by signals from thymic cells and cytokines, they migrate through the thymus. Undergoing positive selection, can they recognize self-MHC? And negative selection, do they react too strongly to self-peptide plus MHC? AR protein in the medulla is crucial for negative selection, forcing expression of tissue-specific self-antigens to weed out self-reactive T cells.
And B cells in the bone marrow.
They develop there from stem cells through distinct stages, pro-B, pre-B, immature, mature, involving sequential gene rearrangement, heavy chain first, then light chain, regulated by transcription factors like PAX5 and cytokines. Allelic exclusion ensures one antibody specificity per cell. Immature B cells expressing surface IgM undergo negative selection against self-antigens. Mature naive B cells, IgM plus IgD plus leave the marrow. In the periphery, B2 cells, the main type, need T cell help for activation, class switching, IgM to IgA, somatic hypermutation, affinity refinement, and differentiation into plasma cells or memory cells. B1 cells are a distinct subset, often T-independent, making natural IgM.
Precise generation and education. But pathogens fight back. What are some evasion strategies?
Oh, they're incredibly clever. Antigenic variation, just changing their surface coats, is huge for viruses like flu and bacteria like Neisseria. Bacteria use capsules to avoid phagocytosis, enzymes to degrade components like C5a, ways to resist opsonization. Viruses establish latency like herpes, block antigen presentation CMV, mimic host molecules. Intracellular bacteria can neutralize phagosome pH, TB, block fusion with lysosomes, or escape into the cytoplasm, Listeria. Defenses vary. Antibody versus cell-mediated depends on the bug. Parasites are tough, often needs combos like antibodies plus eosinophils for worms.
A constant arms race. How do vaccines leverage our adaptive immunity?
Vaccines give the immune system a safe preview of the pathogen or its key components. This primes the adaptive response, generating memory cells without causing disease. Examples include using detoxified bacterial toxins, toxoids like tetanus, diphtheria, often with adjuvants like aluminum salts to boost the response, or using purified viral subunits like the hepatitis B surface antigen produced by recombinant DNA tech, the first anti-cancer vaccine. Newer methods like reverse vaccinology use pathogen genomes to predict good vaccine antigens like for meningitis B.
A safe practice run to build memory. But what if the immune system fails? What are immunodeficiencies?
Immunodeficiencies happen when parts of the immune system are missing or defective, leading to increased susceptibility to infections, often by opportunistic microbes. Primary ones are genetic affecting B cells, like XLA or CVA, leading to bacterial infections. T cells, or both, SCID, caused by various defects like ADA deficiency or problems with T cell receptor signaling. Acquired immunodeficiency is famously caused by HIV, which destroys CD4 plus T cells, progressively weakening immunity and leading to AIDS. Developing an HIV vaccine is tough due to viral variability and latency.
Weakened defenses. What about the opposite problem, overreaction or hypersensitivity?
Hypersensitivity is an exaggerated or inappropriate immune response causing tissue damage. Type I is immediate allergy IgE on mast cells meets allergen, pollen, food, triggering histamine release, hay fever, anaphylaxis. Asthma involves chronic type I inflammation. Graft rejection is hypersensitivity to foreign tissues, hyperacute, preformed antibodies. Acute T-cell mediated against allo-MHC, chronic complex mix. Immunosuppressive drugs, cyclosporine, tacrolimus, rapamycin, are used to prevent rejection but have side effects. Graft versus host disease, GVHD, is the flip side in stem cell transplants where donor T-cells attack the recipient.
Immune system causing harm. What about its role in cancer and autoimmunity?
It's complex. The immune system performs immune surveillance, killing potential cancer cells via cytotoxic T cells and NK cells. But tumors evolve to evade this and can even recruit immune cells like tumor-associated macrophages, TSAMs, to create an immunosuppressive microenvironment that helps them grow and spread. Cancer immunotherapy aims to boost the anti-tumor response, using checkpoint inhibitors, anti-CTLA-4, anti-PD-1, to release the brakes on T cells, or using therapeutic antibodies against tumor antigens, HER2, CD20, or factors like VEGF.
And autoimmunity.
That's when the immune system loses self-tolerance and attacks the body's own tissues. Most are polygenic, many genes plus environment, like type 1 diabetes or rheumatoid arthritis, RA. Some are monogenic, ARI-FOXP3 mutations. Examples include systemic lupus erythematosus, SLE, with autoantibodies against nuclear components causing immune complex deposition and organ damage, RA with inflammatory destruction of joints, Hashimoto's thyroiditis, a T-cell attack on the thyroid. Measuring autoantibodies is key for diagnosis.
Okay, let's take a moment to unpack the vast amount we've covered in this deep dive. We've explored how your body has two fundamental defense arms: the rapid, general innate response, and the slower, highly specific adaptive response. We looked at the innate system's detectors, its weapons, and the crucial roles of antibodies and T-cell receptors in adaptive recognition and memory. We also touched on how this finely tuned system can sometimes malfunction, leading to hypersensitivity, immunodeficiency, or autoimmunity.
And here's where it gets really interesting for you, listening. Understanding these fundamental principles gives you a framework for grasping so much about health: why vaccines work, why some people get sick more often, the basis of autoimmune diseases. The sheer intricacy and coordination are just remarkable.
So what does all this mean for you day to day? Hopefully this deep dive has given you a clearer picture of that constant silent battle your body wages to keep you healthy. The next time you hear about cytokines or T cells or antibodies in the news, you'll have a much better handle on their vital roles.
And this all raises a really important question as we look forward. Given this complexity, this constant learning within the immune system, what are the most promising future directions for harnessing its power therapeutically? Especially against those really tough challenges like persistent infections, complex cancers, maybe even reversing autoimmunity. It's a field just brimming with potential.
Thank you for joining us for this deep dive into the fascinating world of immunology.